Multicellular tumor spheroid models are widely used in drug screening applications. While multicellular tumor spheroids better mimic in vivo conditions compared to TCP-grown monolayers, they lack cell matrix interactions, which would result in a more physiologically relevant model. In this study, we looked into whether the presence and composition of an external matrix would have an effect on spheroid properties. Enzymatically degradable and RGDS functionalized hydrogels were used to mimic cellular ECM. To form the multicellular tumor spheroids, U87 glioblastoma cells were spun down into square pyramidal molds and incubated for 24 hours to allow for spheroid formation. Once formed, the spheroids were encapsulated into non-degradable non-adhesive (nDNA), degradable non-adhesive (DNA), and degradable adhesive (DA) gels, with composition shown in Figure 2. Spheroid characteristics were measured over the course of 10 days. All conditions showed high cell viability for 10 days. In the degradable gels, cells were able to infiltrate into the matrix by day 7 for DNA and day 3 for DA. Since cells were able to remodel the surrounding matrix, the spheroids in DNA and DA became more circular over time and increased in diameter. To measure proliferation, spheroid models were incubated with BRDU for 24 hours and imaged via antibody staining. Proliferating cells were present in all samples, primarily in the outermost region of the spheroids, indicating a proliferative outer layer. Antibody staining for stem cell markers nested in SOX2 was used to measure stemness of spheroid models. In free-floating samples, SOX2 is highly localized to the nucleus, but not as localized in the hydrogel-grown spheroid models. This lack of localization could be due to an overexpression of SOX2 and differentiation of cells infiltrating into the matrix. Finally, spheroid responsiveness to temozolomide was measured at 48 hours exposure to 2 millimolar TMZ. In all conditions without TMZ, we saw viability greater than 90%. Cell viability in free-floating and nDNA models was close to 50% viability when exposed to 2 millimolar TMZ, while the spheroid models in degradable gels showed viability closer to 75%. This decreased response shows that cell matrix interactions affect spheroid model drug responsiveness. In conclusion, we showed that the multicellular tumor spheroid models we created were viable and proliferative for up to 10 days in culture, showed stemness, and had a decreased drug responsiveness compared to multicellular tumor spheroids without cell matrix interactions. For future studies, we want to investigate the presence of a hypoxic core, measure secreted ECM proteins, and utilize PDX cell lines to create models.